Here is something happening outside your window right now that you have never once stopped to find remarkable. A tree is standing in the cold. Maybe it's November. Maybe it's January. Maybe the temperature dropped below freezing 3 nights ago, and the ground has that hard, locked quality that footsteps sound different on. The tree has no shelter. It has no fur, no warm blood, no ability to migrate somewhere more sensible. It cannot shiver. It cannot curl up. It is, by every metric of biological vulnerability, completely exposed to conditions that would kill an unprotected human being within hours. And it's fine. Not just surviving in some marginal, barely hanging on sense, fine. Genuinely, structurally fine. In possession of biological machinery so sophisticated that it reads the coming of winter, weeks before winter arrives, executes a coordinated physiological program across millions of cells simultaneously, survives temperatures that would shatter its tissues if it weren't doing everything exactly right, and then, when the conditions reverse, wakes back up and does the whole thing again as though nothing happened. This happens every year. Every single year for the entire lifespan of the tree, which for some species means centuries. An oak tree that has been doing this for 400 years has cheated winter 400 times. A bristlecone pine in the White Mountains of California has been running this program for over 4,000 years, through ice ages and droughts and conditions that eliminated entire ecosystems around it, and it's still out there right now, still reading the light, still running the program. The word cheating in the title is deliberately chosen and precisely accurate. Winter is trying to kill trees. It's not metaphorical. Freezing temperatures, ice crystal formation, water stress, cellular damage, the complete shutdown of the energy systems that keep living tissue alive. These are real threats. Trees don't avoid them through luck or through being somehow exempt from the physics of cold. They survive by actively countering each threat with a specific biological response, timed precisely, executed across an organism that has no brain and no nervous system to coordinate it. That's the story. Here's how it actually works. Start with the fundamental problem, because the enemy is more specific than just cold. The specific threat that winter poses to plant tissue is ice. Not the presence of cold per se, but what happens at the cellular level when water freezes. Living cells are filled with water. That water, in the warm months, is doing biological work, carrying dissolved nutrients, enabling chemical reactions, maintaining the turgor pressure that keeps cells structurally rigid and functional. When temperatures drop below freezing, that water wants to become ice. And ice is the problem, because ice crystals have sharp edges and expanding volume. Ice forming inside a cell punctures the cell membrane, ruptures organelles, and destroys the internal architecture that makes the cell a living thing rather than a bag of dissolved chemicals. A cell killed by internal ice formation is dead in a way that doesn't recover when temperatures rise. The damage is structural and permanent. This is the primary threat: intracellular ice formation. And deciduous trees, the ones that go bare in winter and return in spring, counter it with a strategy of extraordinary preemptive sophistication. The process begins not when winter arrives, but weeks before. Triggered by something that has nothing to do with temperature. It's triggered by light, specifically by the shortening of days as autumn progresses, the decreasing photoperiod, measured with remarkable precision by photoreceptor proteins in leaf cells called phytochromes. These proteins detect the ratio of red to far red light, which shifts as day length changes, and they transduce that information into chemical signals that initiate what researchers call cold acclamation. The tree reads the calendar with its leaves, not by counting days, nothing as literal as that, but by continuously measuring the light environment with molecular precision, comparing it to an internal reference, and detecting when the change has crossed a threshold that historically has predicted incoming cold. The phytochrome system is essentially a photoperiod detector accurate enough to distinguish day-length differences of a few minutes, refined by millions of years of natural selection in environments where mistiming winter preparation was fatal. When the signal registers, When the phytochrome system indicates that days have shortened past the critical threshold for this species in this latitude, the tree begins a cascade of gene expression changes. Hundreds of genes that were inactive or minimally expressed during the growing season are upregulated. Other genes are downregulated. The molecular character of the tree's cells begins shifting, preparing for a physiological state so different from summer function that some researchers describe winter dormancy as a fundamentally different mode of existence. One of the most critical early responses is the movement of water. Remember that the threat is ice inside cells. The tree's response is to move water out of cells into the spaces between cells, the apoplast, where ice can form without rupturing cellular membranes. This dehydration of the intracellular space is actively managed through aquaporin channels, protein channels in cell membranes that regulate water movement, and it changes the composition of what remains inside the cell. What remains inside is increasingly concentrated with solutes, sugars, amino acids, specific proteins. These solutes are the second layer of cold defense, and they work through a mechanism called freezing point depression. Dissolved substances lower the freezing point of water. Concentrated sugar solutions don't freeze at 0 degrees Celsius the way pure water does. The more concentrated the cellular contents, the lower the temperature at which ice can form inside the cell. Trees actively accumulate cryoprotective solutes during cold acclimation, essentially turning their cellular contents into a biological antifreeze. The sugars involved are not incidentally present, they're partially the product of strategic chlorophyll breakdown. When deciduous trees begin dismantling their leaves in autumn, they're not simply letting leaves die. They're running a controlled recycling operation. Chlorophyll molecules are broken down, and the nitrogen they contain, a precious nutrient, is actively transported out of the leaf and into the woody tissue of branches and trunk for winter storage. The green color fades from leaves as chlorophyll is removed. What remains are carotenoid pigments, the yellows and oranges that were present all along, masked by green, and are now revealed. The reds and purples are something different, anthocyanins that are actively synthesized during senescence, whose function is still somewhat debated, but which may act as a sunscreen protecting the leaf tissue during the chlorophyll dismantling process. The autumn color show is not a beautiful accident of leaves dying. It's the visible signature of a sophisticated nutrient recovery operation, and the colors are essentially industrial exhaust, the visual byproduct of biochemical processes serving specific functional purposes. This does not make autumn less beautiful. It makes it more interesting, which is better. Once the water has moved out of cells and the solutes have concentrated inside, the tree is not done. Because even with the intracellular antifreeze strategy, sufficiently cold temperatures will eventually cause problems. And cold doesn't stop at the temperature where this first layer of defense becomes insufficient. This is where trees deploy something that is, considering the source organism, almost startlingly sophisticated, cold-regulated proteins. During cold acclimation, trees upregulate the production of specific proteins whose function is to manage ice in the extracellular space, to control how ice crystals form, to prevent those crystals from growing large enough to damage surrounding tissue, and to stabilize membranes at temperatures where membranes would otherwise lose structural integrity. Antifreeze proteins, yes, actual biological antifreeze proteins, the same general category of protein that allows Arctic fish to survive in sub-zero seawater, are produced in cold acclimated woody plants. These proteins bind to ice crystals and modify their growth, preventing the formation of large, damaging crystal structures. They are temperature responsive and are expressed in significantly higher amounts as temperatures drop, meaning the tree is continuously adjusting its molecular defenses as conditions change. Dehydrins are another class of cold response protein produced in large quantities during cold acclimation that appear to stabilize cellular structures, particularly membranes, during the stress of dehydration and freezing. They are intrinsically disordered proteins, meaning they don't have a fixed three-dimensional structure in the normal sense, which allows them to interact with and protect a wide range of cellular components. Dehydrin accumulation is so reliably associated with cold hardening in plants that their presence is used as a biochemical marker for cold acclimation status. The tree is manufacturing molecular bubble wrap for its cell membranes. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. The membrane changes themselves are critical and often underappreciated. Cell membranes are composed of phospholipid bilayers, and the fluidity of those membranes, their ability to flex and function rather than becoming rigid and brittle, is temperature dependent. At cold temperatures, membranes made of the same lipid composition as summer membranes would become inflexible and lose function. Trees counter this by actively changing the lipid composition of their membranes during cold acclimation, increasing the proportion of unsaturated fatty acids in membrane phospholipids. Unsaturated fatty acids maintain membrane fluidity at lower temperatures than saturated fatty acids do. The tree is essentially changing its own oil to a winter-grade formulation. All of this is happening in parallel. The water movement, the solute accumulation, the protein production, the membrane remodeling, coordinated across every cell in the tree, driven by the chemical signals cascading from that initial photoperiod detection in the leaves. The tree has no central nervous system. It has no brain to orchestrate this. The coordination is emergent from chemical signaling moving through tissue, from gene regulatory networks responding to shared molecular cues, from the distributed intelligence of millions of cells, each reading the same signals and executing similar programs simultaneously. The result, after weeks of cold acclimation, is a tree that can withstand temperatures dramatically below freezing without dying. Fully hardened temperate trees can survive temperatures of -40 degrees Celsius or lower. Boreal forest species that must contend with the genuinely extreme winters of northern Canada and Siberia can survive temperatures below -60. These are not the temperatures of a moderately cold night. These are temperatures at which metal becomes brittle and rubber cracks, and most biological tissue is simply destroyed. The trees are fine, but surviving tissue death is only part of the winter challenge. There's also the matter of water. In winter, water is locked in ice, in the soil, in the snowpack, in the frozen ground. Trees cannot access it. The physiological water stress of winter is comparable to drought, and a tree that continued operating at summer metabolic rates through winter would desiccate, it would lose water through transpiration without being able to replace it, and it would die of thirst in the middle of all that frozen water. Deciduous trees resolve this problem with an elegance so obvious in retrospect that it seems almost too simple. They drop their leaves. Leaves are where transpiration happens. Leaves are where the enormous surface area for water evaporation exists. Remove the leaves, remove the transpiration, eliminate the water loss problem for the duration of winter. The leaf drop is not incidental to autumn. It is the critical water management strategy, timed by the same photoperiod detection system that initiates cold hardening. The mechanism of leaf drop involves the formation of an abscission zone at the base of each leaf stalk, a layer of specialized cells that weakens controllably until the leaf detaches. The abscission zone formation is regulated by the changing balance of plant hormones during senescence, particularly the decrease in auxin, which suppresses abscission, and changes in ethylene, which promotes it. The leaf doesn't fall randomly when it dies. It detaches at a specific preformed location, leaving behind a sealed scar that protects the twig from pathogen entry and water loss. Beneath winter's surface, below the frost line in the soil, the roots of deciduous trees are still alive and still conducting limited functions. Root cells undergo their own version of cold acclimation, calibrated to the more moderate temperatures found in soil compared to air. Some water uptake, and limited transport continues at very low rates. The tree is not entirely metabolically shut down in winter. It is more accurately described as operating at minimal life support levels, maintaining basic cellular integrity, and the slow biochemical processes that will allow rapid resumption of activity when conditions change. The buds on winter trees are another level of the cheating death architecture that deserves specific attention. Those small, hard structures at branch tips and leaf axils are not dormant nothing. They are next year's leaves and flowers wrapped in protective bud scales, modified leaves impregnated with resins, waxes, and other waterproofing compounds that maintain the delicate meristematic tissue inside at temperatures that would kill unprotected cells. The bud scales are the winter coat for the tree's most vulnerable growing tissue. Inside those bud scales, the future leaves exist in miniaturized form, paused at an early developmental stage, protected and waiting. When spring comes, the activation of those buds represents not the beginning of new growth from scratch, but the resumption of development that was interrupted and preserved across winter. The tree didn't plan next year's leaves in spring. It planned them in the summer before, packaged them in protective wrapping in autumn, and stored them through winter for spring deployment. Now discuss spring, because the return from dormancy is as sophisticated as the entry into it, and it requires a different but equally remarkable set of mechanisms. Trees do not simply resume when temperatures rise. If they did, they would risk catastrophic miscalibration. A warm spell in January followed by a return to freezing could kill reactivated tissue that had abandoned its winter defenses prematurely. A tree that leafed out in a January warm spell and then experienced February frost would be in serious trouble. The risk of being fooled by temporary warmth is real, and trees have evolved a remarkable solution to it. They count cold, the chilling requirement, sometimes called vernalization in a related context, is the mechanism by which trees accumulate cold units across winter, registering how much cold exposure they have experienced, and requiring a specific minimum threshold of cold before they become capable of responding to warmth. A tree that hasn't received sufficient chilling cannot break dormancy even when temperatures are favorable. It is locked in dormancy until the cold requirement is satisfied. This is not a simple binary. It's a quantitative accumulation system, counting hours of exposure at temperatures within a specific range, typically between 0 and 10 degrees Celsius, across weeks and months of winter. Only once the accumulated chilling hours exceed the species-specific threshold does the tree shift from endodormancy, dormancy maintained by internal signals regardless of external conditions, to ecodormancy, dormancy maintained only by unfavorable external conditions. After that threshold is crossed, the tree is primed to respond to warmth. Accumulation of heat units above a threshold temperature then drives the progression from ecodormancy to bud break. The two requirements, sufficient cold accumulated, then sufficient warmth, form a two-factor timing system of extraordinary precision. The reason this is immediately important beyond its biological elegance. Climate change is disrupting the chilling requirement system in ways that are visible in the ecological record. As winters warm, some species and some regions are not accumulating sufficient chilling hours. And the timing of spring bud break is shifting in ways that create mismatches with other ecological events. When trees leaf out at different times than the insects that depend on spring leaf emergence, and when those insects hatch at different times than the birds that depend on them, the cascading effects of phenological disruption spread through ecosystems in ways that are still being mapped. The tree's winter timing system evolved over millions of years to match the reliable rhythm of temperate seasons. When that rhythm changes faster than evolutionary adaptation can follow, the system begins producing errors. The same sophistication that allowed precise seasonal synchrony becomes a vulnerability when the seasonal signal it was calibrated to read starts shifting. Before finishing with deciduous trees, pause for evergreens because they are doing something categorically different and equally remarkable. Conifers, pines, spruces, firs, larches in the deciduous exception keep their needles through winter. They don't abandon the leaf surface area. They maintain it. This sounds like the harder strategy, and in some ways it is, which means the mechanisms they've developed to make it work are even more impressive. Conifer needles are leaves engineered for winter retention. They're narrow, reducing surface area and wind exposure compared to broad leaves. They're coated in thick, waxy cuticle that dramatically reduces water loss through the needle surface. The stomata, the pores through which gas exchange happens, are recessed into grooves, further reducing transpiration. The cells inside the needle contain higher concentrations of antifreeze compounds than deciduous cells need to maintain in summer. The needle is a winter-hardened leaf, designed from its developmental inception to withstand conditions that would destroy a maple leaf. Conifer needles still undergo cold acclimation in autumn. They still upregulate antifreeze proteins and cryoprotective solutes and modify membrane lipid composition. But they do so while maintaining the needle structure that allows limited photosynthesis to continue even in winter on sunny days when temperatures are above freezing. The evergreen strategy accepts the metabolic and water stress of maintaining leaf tissue through winter in exchange for the capacity to photosynthesize whenever conditions permit, including during the early weeks of spring before deciduous trees have rebuilt their leaf canopy. Conifers dominate boreal forests partly because the combination of winter-hardened needles and early spring photosynthesis gives them an advantage at high latitudes, where the growing season is short and every photosynthetic day counts. The dark green of a pine forest against winter snow is not the color of something struggling to survive. It's the color of a different strategy winning in a different context, a strategy refined over roughly 300 million years of conifer evolution, in environments ranging from tropical coasts to the tree line at high altitude and high latitude. Here is where the full picture becomes something worth sitting with because the biological engineering described across this entire account, the photoperiod detection, the gene expression cascades, the membrane remodeling, the antifreeze protein production, the chilling hour accumulation, the bud scale architecture, the abscission zone. All of it is executed by organisms with no brain, no nervous system, no muscles, no ability to sense their situation the way an animal senses its situation. Trees are doing all of this through the distributed chemistry of cells responding to molecular signals, through gene regulatory networks encoding responses selected over geological timescales, through the slow physics of water potential gradients and membrane permeability. The sophistication is not the product of intelligence in any conventional sense. It's the product of evolutionary time, of the brutal selection pressure of millions of winters eliminating every tree lineage that didn't get the molecular details right and preserving and elaborating the ones that did. The tree outside your window isn't thinking about winter. It isn't bracing itself or deciding anything. It's running a program written in the language of biochemistry by 400 million years of evolutionary pressure. A program that every winter has been field tested against the actual conditions that evolution was written by. It always passes the test. That is not nothing. That is, if the word is appropriate for something that has no awareness of its own achievement, triumph. The quiet, bare, apparently dormant tree in the frozen ground is one of the most sophisticated biological systems on Earth, running one of the most elegantly engineered survival programs in the history of life. It just doesn't look like it, which is part of what makes it worth understanding. Every winter, every year, every tree, the cheat runs, and every spring it works.